The second speaker of this opening session is not less provocative and visionary. It's Professor Takema Fukatsu from Tsukuba. Takema is a professor at Tsukuba University. He's a senior researcher at the National Academy Institute, but he's most known for being the director of something similar as a CRC here, and that is from Japanese government-funded ERATO project, and it's known as the Fukatsu Evolving Symbiosis project. And when following him for the years, it's just an amazing story and fits so nicely now to Tom Gilbert, because Tom raised this issue of domestication. But to make from a wolf to a dog, it takes a few months or weeks or years. And Takema will pick up on that issue and talk about experimental evolution of Insect-E.coli Mutualism. Takema.
Okay, thank you for the kind introduction, Thomas. And hello, everybody. I'm Takema Fukatsu from AIST, Tsukuba, Japan. And first of all, I'd like to express my sincere gratitude to Thomas for inviting me to this exciting opportunity workshop. And this is my first time to visit Germany, and except for the flight stopover. So now I'd like to enjoy this opportunity. And Thomas is truly a nice guy, my good friend. This is a smash shot at the Golden Reach Conference in Italy in June. And this is the smash shot the last evening. Thomas kindly invited me to the dinner at his home. Thank you for the hospitality. And this is a thank you for your kids come to Japan, and he's enjoying the Japanese sake like this. Anyway, okay, I'll go to my talk. I will talk about the experimental evolution of Insect-E.coli Mutualism. Introduction. In nature, microbial symbiosis is universally important. The essence is acquisition of peculiar and efficient biological function microbes enables exploitation of novel food resources and ecological niches. And insects account for the majority of macroscopic biodiversity described so far. And diverse insects have elaborate symbiotic systems for harboring specific and beneficial microbes. There are diverse associations between insects and microbes, ranging from parasitism through commensalism to mutualism like this. Our previous studies are widely and intensively investigating a variety of symbiotic associations between diverse insects and microbes, including novel biological functions and phenotypes conferred by microbial symbionts. Evolutionary origins, processes, and dynamics of intimate host-symbiont associations. And understanding of microbial symbionts relevant to agriculture and medical pests. These are examples of obligate endocellular symbionts within bacteriocytes of aphids and tsetse flies, and obligate extracellular gut symbionts within the midgut symbiotic organ of stink bugs. In this way, host and symbiont are metabolically integrated into an almost inseparable biological entity. And here is a fundamental question. Where have these obligate symbionts come from? Obligate interface bacterial mutualism is among the most sophisticated forms of symbiosis. However, such obligate symbiotic bacteria must have been originally derived from free-living environmental bacteria. Then how have highly specialized obligate mutualists arisen from less specialized free-living microorganisms? How to answer this question? And recently, experimental evolutionary approaches to symbiosis have emerged like this. And we established a novel Insect-E.coli experimental symbiotic system. This is derived from Takahiro Hosokawa's amazing work. This is a blind-winged green stink bug, Protea starry. This this insect, when dissected, this is the alimentary tract. And the posterior end of the alimentary tract is yellow in color and specialized. It consists of numerous small crypts, and thousands, hundreds of crypts are arranged in rows and inside full of symbiotic bacteria. That is essential for their survival and growth. And these symbionts are vertically transmitted from mother to offspring. Mother smears the bacteria on the eggshell, and then babies now passionately sucking the symbiotic bacteria smeared on the eggshell and acquire it. If it fail to acquire the bacteria, they cannot grow. And we identified 6 distinct lineages of enteric gamma protein bacteria. Here I tentatively called symbiont A, B, C, D, E, and F, and from Japanese populations of this insect. And this is the geographic distribution of the symbiotic bacteria. And interestingly, symbiont A is fixed across the mainland population. And on the other hand, and in the southwestern island of Japan, symbiont B, C, D, E, and F are coexisting like this. A is fixed in the mainland population, while in southwestern islands, B is major, while we are minor C, D, E, and F coexisting. So indicating remarkable symbiont polymorphism between and within local population of this insect. And while the major symbionts A and B cannot be cultured, minor symbionts C, D, E, and F are easily cultivable even on the LB agar plates. And these are the complete genomes of the symbionts A, B, C, D, E, and F. And like this, uncultivable major symbionts A and B exhibit degenerative genomes in contrast to large genomes of cultivable minor symbionts C, D, E, and F. Then, symbionts A, B, C, D, E, and F are all essential for the host? The answer is yes. All the A, B, C, D, E, and F are regarded as obligate mutualists. When get infected, the insects can grow. Without it, it will all die. Then, what will happen when the original symbiont B is replaced by the different one? For example, here, this original symbiont B infected egg are surface sterilized by formalin and alcohol, just washing the egg. Easily, symbionts are removed. Then, newborn nymphs become aseptic. Then, these babies are fed with cultured symbiotic bacteria, like under this setting. And then, their symbionts are completely replaced by the new one. And they all grow, looks healthy. So it indicates that the symbiote replacement worked, and normal, healthy green stem bag emerged like this. So consequently, Proteus stari, this stink bug, is an excellent model for insect symbiotic studies. Quite an experimentally tractable, and within the species, obligate symbionts and cultivable symbionts, different level of the symbiotic evolutionary stages coexist. That it really this is in the process of symbiotic evolution in the wild, in the natural setting. But anyway, and furthermore, the symbionts A to F are related to enteric bacteria. They belong to the genus Pantoea. E. coli, Escherichia coli, is here. They're not pretty closely related to them. Then what will happen? Proteus stari is experimentally infected with the model bacterium E. coli. And we did it. And this is the normal insect infected with the original symbiont. Green, large in size. Adult emergence rate is over 80%. And when the original symbiont was replaced by E. coli, most of them die as nymphs. But small number, from 5 to 10% of the insects manage to become adult, but adult insect looks like this, small in size. Then the color is strange, brown colored. And when these normal insects are dissected, symbiotic organ is well developed and yellow in color, and in this insect, very thin, colorless. It almost looks empty. But when we do fish, not empty at all. E. coli are really localized to the exactly correct place for the symbiont localization like this. And some of these tiny insects managed to mate and lay eggs. Then on the egg surface, E. coli was smeared. And newborn nymphs can acquire E. coli from the egg surface like this. So it means that, although incompletely, E. coli can establish infection and support survival of Proteus-staurii. Then, experimental evolutionary approach may be feasible. By inoculation and continuous maintenance of E. coli in this insect, would E. coli evolve into a symbiotic-like entity? So, we designed an artificial evolutionary experiment using 100 times faster evolving E. coli. Using hypermutating E. coli strain delta MAT-S, in which DNA mismatch repair gene, enzyme gene MAT-S is disrupted. The E. coli shows a very fast evolutionary rate. And so here the evolution is accelerated really by 100 times. Then such an E. coli is infected to the insect and maintained from generation to generation like this. Then what will happen? Then unexpectedly, in multiple evolutionary lines, the initial evolutionary rate, emergence rate is less than 10%, but some of them rapidly improved to over 30% like this. And in such evolutionary lines, colony shape of E. coli changed to small and convex like this. And the host became larger and greenish in this way. So in this way, we established a novel experimental model system for symbiotic evolution. Experimental evolution of E. coli into a symbiotic bacterium in laboratory, and complete description, analysis, and investigation of the evolutionary process will lead to an ultimate understanding of symbiosis. And Ryuichi Koga, him, successfully made E. coli evolve into insect mutualists in the laboratory. When inoculated to Proteus-staurii, all standard laboratory E. coli strains, these strains, showed similar phenotypic effects, very low adult emergence rate, dark body color, and small body size. So we selected BW25113 as a background wild-type E. coli strain. And this is the evolutionary setting. BW25113 background wild-type strain, delta MAT-S. Here, its DNA mismatch repair gene MAT-S is disrupted. This is a non-mutating control strain. Its phage integrase gene is disrupted, but it has no phenotypic effects. And in growth selection, the first emerged adult insect was subjected to dissection of symbiotic organ for inoculation to the next generation, as well as freeze storing. And in color selection, the most greenish insect was subjected to dissection of symbiotic organ for inoculation to the next generation, as well as freeze storing. And in this way, we ran experimental evolutionary lines for 10 to 14 host generations over 2 years. Then we established and maintained 12 CML color selection lines with 11 CIL control lines and 7 GML growth selection lines with 7 GIL control lines like this. And notably, in a color selection line, CML05, adult emergence rate here jumped up at generation 7, and the high emergence rate were maintained subsequently. And in a growth selection line, GML07, adult emergence rate jumped up, improved as early as generation 2, and subsequently the high rate was maintained. And hereafter, we focused on these improved lines and unimproved lines for the lines and generations. And here are body color and colony shape and emergence rate. in the improved lines, the coincident with the improvement of adult emergence rates, body color of adult insects improved from dark to greenish. And the colony morphology of E. coli changed from large and flat with rich extracellular matrix to small and convex with a little extracellular matrix. And in addition, the improved evolutionary E. coli lines exhibited distinct microbiological traits in comparison with the original E. coli, including reduced growth rate in the liquid media, and strikingly smaller cell size, and loss of flagellar motility. These are rainbow plots of moving trajectory for 2 seconds, and swimming ratios, no movement. And this is the transcriptomic analysis of the improved and non-improved evolutionary lines. And interestingly, the gene expression pattern of E. coli after the improvement of host phenotypes were separately, very beautifully separately clustered from those without the improvement. And these results suggest that the evolution of the mutualistic E. coli entails specific and global change of gene expression patterns. And then we determined deeply covered high-quality genome sequences of the improved lines and non-improved lines throughout the evolutionary course. And I identified over 1,000 detected variants and over 200 fixed variants. And of these, we identified 7 and 9 candidate mutations for CML05 and GML07 respectively that are correlated with the improvement of the host fitness. And of these, we focused on this SIA mutation in CML07 and this one CRP mutation in GML07. These are the candidate mutations of interest. In CML05, a gene encoding adenylated cyclase, SIA, is disrupted by a frame shift mutation. And in GML07, a gene encoding cyclic MP receptor protein, CRP, has a substitution here that from leucine to proline at the functionally important cyclic MP binding site. And interestingly, they constitute the common bacterial global metabolic regulator system, carbon catabolite repression or CCR pathway. And we introduced these mutations of SIA and CRP to wild type E. coli genetic background, like this. Then the colonies of the E. coli became small and white, like this. And cell size becomes smaller and stop moving. And when they were infected to the host insect, adult emergence rate are drastically elevated and they become larger and greenish. So indicating that the single disruptive mutations of the CCR pathway gene make E. coli an insect mutualist. Even a single mutation is sufficient for evolution of the mutualism. So conclusion, single mutations on the CCR pathway quickly and convergently occurred to make E. coli mutualistic to Proteus stari. And so evolution of mutualism may be easier than conventionally envisaged. And this may account for why mutualistic symbiosis are universally found in insects and other organisms. And these studies are conducted under the Thomas kindly introduced under the ERATO project. This is a project scheme, and ERATO original file cases with the project leaflet are available at the desk behind. So please take one free. And they are the great collaborator for the project and fundings. And thank you for your attention, and any questions are welcome. Thank you.